around for the entire presentation. So welcome to our second workshop that is sponsored by or provided by the GUIDES shared resource within the Native American Cancer Prevention Partnership. So today it is my honor to introduce Greg Caporaso, who's an associate professor in biological sciences and the computer science departments at NAU. He also is a director of the Center for Applied Microbiome Science in the Pathogen and Microbiome Institute. His area of expertise is in microbiome bioinformatics, the human gut microbiome, and software engineering to support biological research. He was first author and primary developer of the CHIME, and I see you actually wrote it out for me, microbiome bioinformatics platform, which has been cited over 25,000 times since its publication, as well as many other popular bioinformatics software tools. He's also the author of the Sloan Foundation funded free bioinformatics text, An Introduction to Applied Bioinformatics. So we're really honored to have him here leading this workshop. We hope that it is interactive. What I suggest is, it looks like most everyone but Ron and I have our cameras off, which I think is helpful in terms of bandwidth. But if you have questions and you really want to remember those questions, if you could put it in the chat, and then I will help Greg at the end sort of navigate those questions. I think that might work pretty well. With that, I'm going to hand it off to Greg, and then we will hopefully be able to see his slides sooner than later. So Greg, with that, I'm going to mute myself and let you go forward. Thanks.
Great. Thanks so much, Janie. And if we get a few minutes in and I can't share, or I am not able to share my screen, I can also just take a minute and share a link to my slides, and folks can then pull it up on their computer. Yeah, thanks for the introduction, Janie, and thanks everybody for your time. My goal with this presentation today, bioinformatics is a big field. It's a diverse field, very cross-disciplinary. And so when I started thinking about how to do a 1-hour workshop, I figured that the best thing I could do would be to share some ideas with you on how you can integrate bioinformatics in your research, or steps that you could take to do that most successfully. My goal with this presentation is to really introduce the field at a very high level. I'm also happy to take questions at any point. If questions come in, you can feel free to interrupt me with those, because I'd like this to be as relevant as possible to you. And if I don't get through all my slides because we get bogged down with questions, that's totally fine with me. So as I'm sure you're aware, biology has very rapidly become a data-intensive field. In large part, this has been driven by the decreasing cost of DNA sequencing, which ushered in the era of genomics. But since the beginning, since the sequencing of the human genome in 2001, those DNA sequencing technologies have been adapted for many other applications, including microbiome research, which, as Janie mentioned, is my area of expertise, RNA-seq analysis, related tools for understanding transcriptomes and metabolomes and proteomes, and so on. Also within the last decade, mass spectrometry has also been getting more financially accessible. And so that's really driving things like proteomics research and metabolomics research. All of these individual data types, whether it's microbiomes, genomes, proteomes, metabolomes, are challenging to work with on their own because they tend to generate very large data sets. And so it's caused biologists to need to start learning tools that haven't traditionally been in the toolbox. Things like programming languages, command line software, relational databases. And so these are a lot of new skills that biologists are having to learn. And in part, that's a good component of what I work on is making tools accessible to biologists, making advanced computational tools, things that people can learn without being computational experts. Oh, great. So I can share now. So let me try and do that. I will share. Okay, so you should see my slides up now.
Yeah, we see it. Thank you.
Okay, great. Okay, so yeah, so many new tools in the toolkit, and a lot of biologists are scrambling to learn those tools. And there really are starting to be a lot of good resources that can help with learning those tools. And I'll touch on some of them throughout my talk. And as if these individual data types weren't hard enough to work with on their own, the latest challenge on the horizon is what I would refer to as integrated multiomics, where we're taking different data types. So for example, if we have some metatranscriptome data from an RNA-seq analysis, and we have some metabolome data from a mass spec analysis, say all from the same samples, maybe from cases, disease cases and controls, trying to integrate those different data types so that we can learn more about them together than we could learn from any of those different types on their own. And so, for example, in the metatranscriptome metabolome area, we might be trying to learn what genes are being transcribed and how that impacts the small molecule microenvironment in a given type of tissue. And so, in addition now to needing to know how to learn needing to know how to work with, say, RNA-seq data, and needing to know how to work with metabolome data, which are totally different data types, one from mass spec, one from sequencing. Now we need to be thinking about how do we integrate those? How can we, say, develop models that can appropriately deal with data of those two types? So since bioinformatics is basically now touching almost all aspects of biology, I wanted to start my talk today with some tips that I hope will be helpful for researchers who are taking on a project that has a bioinformatics component to it. And then I'll spend some time talking about the bioinformatics work that my lab does. And I'll specifically talk a little bit about the Chime platform that Janny mentioned in the introduction. And then I'll wrap up talking a little bit about open science and why it's important and things that you can do to make your science more open. Again, please feel free to interrupt with any questions that you have as we're going through. And now let me just see about advancing my slides here. Okay. Yeah, so like I said, I'm going to start out with some tips for taking on a new bioinformatics project. And I tried to think about this. I work with a lot of folks who are not bioinformaticians, not computer scientists, but they have a big bioinformatics component of a project. And so maybe they've done some microbiome sequencing or metabolome profiling or a little bit of both. And so I tried to gear these ideas to things that would be helpful for that type of audience. And I'm sort of assuming that that's kind of the situation that most of the folks in the audience here today are in, where they might be needing to use bioinformatics in their work, but not necessarily have programmers or bioinformaticians on their staff. So the first one, you often hear this from, you probably have often heard this from your statisticians, but when you're planning something like a project that's going to involve a lot of data generation, a lot of analysis, it's a good idea to talk to bioinformaticians early on, like in the project planning stage. And this can help you figure out, how to design your experiment, what kind of controls you might need. And so, for example, like in a microbiome survey, we often like to try and include some artificial microbiome samples where we have, we call them mock communities, but where we have like pooled together organisms in known abundances, and we can include those in the sequencing run. And that can often be a useful way to detect any errors that have arisen. And a bioinformatician can also tell you, if software already exists for what you're trying to do, or if new software is going to need to be designed. And whichever of those situations happens to be the case can have a big impact on your budget for the project. And so just like you want to try and talk to a statistician early on in a project, it's also a good idea to think about talking to the people who would advise you on the data analysis early on in the project. Oops, wrong way. So this is another one that I run into a lot. A lot of times when folks have somebody on their team who has some programming experience, it can be tempting to build custom software to carry out a project. But I like to encourage people to use existing software if at all possible. Even if you have somebody who does know how to program, a lot goes into developing a software project for a bioinformatics project. And there can be some aspects of this that are very challenging for folks who haven't actually, say, been trained in software engineering. So software validation, in other words, designing sort of positive and negative controls for your software to make sure that it's doing what it's supposed to be doing, is something that's really important. And it's often something that reviewers are going to want to see if you are using new software in your analysis. And so there often is a temptation for folks to build new software, but it is usually a bad idea if you can avoid it at all. A lot also goes into, if you want to distribute that software, a lot goes into that. And when I talk about my software project, I think that'll give you a good idea of how big of a project it is that you can be taking on when you start developing custom software for a project. When there is existing software, choosing which software is hard, which software to use can be hard. And it's not a bad idea to discuss with a bioinformatician how to make that choice between which existing software you should be using. Things like how well documented, how well validated the software is, are very important. Validation in particular can be challenging for somebody who doesn't have a lot of software experience to evaluate. Another thing is, like, let's say you're doing an RNA-seq project, you may start by, Googling for RNA-seq analysis software, or by looking at, what others have used in papers that you have read recently. One thing that you want to check is, is this still an active project? And there's ways to tell that, by looking at the project's website, looking at their GitHub repositories, which is often where the code will be stored. And you want to basically what you want to figure out is this something that people are actually using in the field today? If there are bugs introduced to the software, is somebody addressing those? Has this been updated to work with the most recent, say, DNA sequencing technologies? If this is, if it's a project that maybe a grad student worked on 4 or 5 years ago and nobody has touched it since, the fields are moving so quickly that it has probably been superseded by something else. And you want to know that as soon as possible, because you're going to invest a lot of time in learning that software. You're going to invest a lot of time in using that software. And if you get to the stage where, your project is in, your paper is in peer review, it can be really discouraging to get feedback that the software you're using is really out of date and you should be using something else. This is a really important one. So metadata is really the key to a bioinformatics project. And so, when we're doing a microbiome project, it's nearly impossible to discern anything about the samples that we're working with if we don't have good metadata. And so like that metadata is basically going to be like just about everything that you can imagine collecting about those samples. And so like if this is, say, a human subjects project, what subject is this from? What group were they in? If this was, say, like a case control, when was the sample collected and processed? Who collected and processed the sample? So in addition to just doing the basic analysis on your project, that metadata can be really essential when you are trying to identify artifacts in the data set. And so I have run into situations where we needed to know, for example, like who did a DNA extraction on these samples because some protocols seem to have differed that were having an impact on the results. And so keeping that information, keeping, say, a spreadsheet or a database of that type of information at the very beginning of your project when you're starting to collect data will really pay off when you get to the analysis stage of your project. There are often also, depending on what field you're working in, there are often metadata standards. And so there's a collection of standards called the mixes standards that are relevant for sequence data. That's it's MIXS. And the X is basically it is minimal information about any sequence data. And then based on like what data type you're working with, that X, that any would be filled in with something different. And so like there's metadata standards for microbiome sequencing projects that differ from metadata standards for RNA-seq projects versus metabolomics projects and so on. And that is going to become important when you get to a stage where you're depositing your data in a long-term archival resource. So for many projects, particularly if they're federally funded, you might have requirements to publish your data in a public repository at the end of the project. It's a good idea when you are starting your project and even before you start collecting your data to understand what the metadata standards are for the field that you're working in, because otherwise you could end up in a situation where maybe 5 years have gone by, you're ready to wrap up this project and deposit the data, and you find out that the standard for this field is to report some piece of information that you may have been relevant to your project 4 years ago, but you haven't thought about since then, and you don't know, you didn't know that you needed to retain that information. And so it's a good idea to figure out what those standards are early on, so that when you get to the data deposition stage, you will make sure that you have everything that is relevant for your data type. Another one is the computational reproducibility is particularly hard for folks who have not been trained in bioinformatics or computer science. And what I mean by that is, we're all used to writing a detailed methods section where we describe what lab protocols we used at a very detailed level, and how we, like say, design the experiment, how we collected samples. But it's also typically expected when you have a bioinformatics component to your project that you're going to provide very detailed information on what analysis steps were run, what software was used, and that can often make or break your work for a given publication venue. And so some journals are going to be very strict that they want to make sure that the bioinformatics steps of this work can be reproduced. And so if you don't have that detailed information, that could prevent you from publishing in that particular journal, say, or it could end up creating a lot of work for you downstream. So if you get some comments back on your paper and they want to know something about your computational workflow that you don't have recorded, it's possible that you could end up having to rerun all of your analyses in order to be able to, say, answer that reviewer question about what information was missing. So the way what I typically tell people to do when they have a bioinformatics component in their project is keep a computational lab notebook. And so this can be like as simple as a Word document or a Google Doc. What I'll often do when I'm starting a big analysis project is I'll create a new Google Doc. And if I'm working with command line software, for example, I will record information about when and where I obtained that software from. So like I downloaded this, say, from the project website on February 24th, 2021. And then let's say this is command line software. I will keep track in that document of every successful command that I ran in that analysis. And I will often put some annotation in there too. And so I'll say, something like, at this stage, I am building a phylogenetic tree from my multiple sequence alignment. And then I'll put that command in there. And just building up those commands sort of acts as your lab notebook. And if you keep that and you're nice and neat with it, you can actually probably just create a PDF of that when you get to the publication stage and submit that as a supplementary methods file along with your paper. And then that will help your reviewers and your readers figure out exactly what you did. But honestly, the person who that tends to help out most often is yourself, or, like in my example, it might help me out when a few months later, I either need to go back and figure out what I did so that, say, I could apply it to a different data set, or if I need to answer a detailed question about the analysis, I can usually look that up in my notes, and I'll be able to answer that question, say if somebody asked me about it at a meeting or something like that. So like this idea of a computational lab notebook doesn't have to be anything very complicated, but it's just the same kind of idea as what you're doing with your wet lab notebook, where you're keeping detailed notes on everything that you're doing. This is something that'll just save you a ton of time and a ton of frustration if you're doing this with your computational work as well. And the last thing I'll mention here is there are tons of great workshops going on these days. A lot of them, including some of my own, have moved online. But if you know that you have a big bioinformatics component of a project coming up, and say you've got a PhD student or a postdoc who's going to be performing that analysis, start thinking about getting them signed up for a workshop. And a great resource for this are the software or data carpentry workshops. And so these, I imagine some folks in here have heard of these already. They're very popular. This group has been doing this for about 10 years now, maybe even more than that, maybe 15 years. And there is probably, they have instructors all over the world. There are a number of instructors based here in Arizona. And so U of A does these, U of A hosts these workshops from time to time. I think NAU has even hosted one or two of these. But the idea is like, this is anywhere from, I think usually from like 2 to 5 days. And they will teach some of these ideas that I'm talking about. So like how to think about ensuring reproducibility of your work, or how to think about managing data and metadata so that it will be most accessible to you, or getting some introductory skills with a programming language like Python or R. And so there is probably like a software data carpentry workshop probably happening like in pre-COVID times, probably, once a week somewhere in the world, they're that popular. And now I think they are doing a lot of online workshops, which makes them even more accessible. Those would be general purpose workshops that would build skills that are relevant for bioinformatics and for data analysis. You can also, like, if you know you're going to be using some specific software, you can see if the team that builds that software offers workshops. And in addition to going and learning about the software that you need to use or picking up some new skills, they also really are often very good networking events, because it's an opportunity to get together with other folks who are using the same software and therefore are probably carrying out similar projects to what you're carrying out. So I hope that these were helpful. And this is sort of a natural break point in my talk here. And so if there are any questions, I would be happy to take one right now. And if not, I can just keep rolling.
So if anyone has any questions, if you want to unmute and ask. I didn't see any in the chat. Okay, all right. Well, keep rolling.
Okay, sounds good. Okay, so the next thing that I was going to do is I was just going to give a little bit of a background on the work that my lab does. And this is primarily, I'm going to talk about our Chain2 microbiome bioinformatics platform. And so I realize not everybody in this workshop is doing microbiome research, but I'll just sort of talk about this generally so that you can get an idea of what an active bioinformatics software project looks like, and what the different components of this are. And so this project is something that I have been working on for about 10 or 11 years now. Chain1, which was the precursor to Chain2, it started as a postdoc project for me. I was working in a microbiome research lab in Colorado, and I had a background both in biology. I had just finished a PhD in biochemistry, and I also, as an undergrad, studied computer science. And so I did, there weren't really bioinformatics programs at the time. And so I sort of worked around that by doing a double major in computer science and biochemistry. And then did a PhD in biochemistry, but it was very bioinformatics focused. And so I ended up joining a microbiome research lab that had been developing some very cutting edge methods, both lab techniques and software. And my role on that project was building out some software so that we could make the methods that were being developed in that lab at the time generally accessible to the microbiome research community. And over time, this has turned into a massive collaborative effort. And so you can see this is the author list for the Chain2 paper, which we published about a year and a half ago now. And we had over 100, I think 110 co-authors on this paper from about 60 different research institutions in about 10 different countries. And so this project is a massive global collaboration to build this software. And what it does, so like initially it was designed to perform a taxonomic and phylogenetic analysis of a microbial community. And so that could be something like a gut sample from a human. If this was a human gut microbiome project, it was designed to support things like a 16S ribosomal RNA survey, or for example, an ITS survey. But as the field has progressed, we have started to think about obtaining other data types to get a more functional view of the microbiome. And so not just who is there, but what are they doing in this environment? What genes are they transcribing? What proteins are they producing? What does the small molecule environment look like based on the metabolome? And so I like to use an analogy here from GIS, Global or Geographic Information Systems. Google Maps is a simple GIS system, just as an example. And with a GIS system, you are trying to model the real world. And you're doing that by thinking in terms of data layers. And so depending on what you want to do, you might need certain layers or you might not need certain layers. And so, for example, if you are, say, planning a hiking trip, you might need different layers of data than if you're planning a construction project. And so just like in GIS systems, you use layers to model the real world. In a microbiome analysis, we think of like these different data types as the different layers. And so metabolomes, proteomes, and transcriptomes are different layers in modeling a microbiome. And so one thing that is really interesting to think about here is a lot of these data types, or a lot of these different data types, they start out needing some different types of processing. And so if you're working with data that's coming from a mass spec instrument, and so like you're doing a metabolomics analysis, and you are also doing an RNA-seq analysis, so you're doing some sequencing data, you're generating some sequencing data. Those are two really different data types at the start. And so in one case, you're dealing with like these peaks from a mass spec instrument, and another you're dealing with sequencing data and associated quality scores. And so the system or the workflows are going to be pretty different in the beginning. Like you're going to have some metabolome processing, you're going to have some RNA-seq processing. But what the commonality is between a lot of these different data types is that you get to some sort of a table where you have samples on one axis and what we usually call features on the other axis. And so like the samples are going to be the samples in your study. So maybe these could be something like if you're comparing cancer and adjacent normal healthy tissue, you might have your cancer group, you might have your healthy tissue group, and then you've got a number of different subjects or samples from each of those. And then the features on that other axis, in your metabolome survey, those might be metabolites that you observe. In your RNA-seq survey, those might be specific transcripts that you observe. But the fact that that table looks the same, that makes it so that a lot of the same analysis workflows can be applied downstream. And so a lot of the things that you can do, say, for example, on a 16S survey, you can adapt to be able to do with metabolomic data and RNA-seq data. And so when you're starting to think about an analysis that you want to do, you can often start looking at what the other like omics fields, what tools exist for these other omics fields. And there may be ways to adapt that. And so that sort of broadens the set of tools that you have available to work with a specific data type. And so where we're going with Chain 2 now is that we are supporting analysis of many of these different data types with the goal of ultimately supporting integrated omics analysis. And so we want to be able to, like I said before, pull the metabolome data and the transcriptome and the proteome data together to build a better model of the microbial system that we're studying. A couple things I'll just go through real quick here. Chain 2 is built on what we call a plugin system. And so that means that we don't necessarily build all the software like on my team directly, but we work with other groups who are building their own software, and we work with them to integrate it into the Chang-2 system. And so in addition to like our core development team, we have a lot of third-party developers. These are often like grad students or postdocs working on specific types of analyses. And we're building this community where folks can share their plugins. And so we have this site that we have a version of it up now. We have a new version going up soon, but it's basically a site for sharing tools for this type of analysis. And so right now it's focused on microbiome analysis tools, because that really is like the primary focus of Chang-2 at the moment. But we are now starting to see things like metabolomics tools being added to this toolkit, and even some RNA-seq tools. And so this could be a good spot, if you have a bioinformatics component to your project, you can feel free to reach out to me and just ask, is this something that like would work well, something that would be possible to do with Chang-2? One of the other cool things that we have really focused on with this system, I mentioned that I am very interested in making these powerful tools accessible to people who are not trained as computer scientists or computer programmers. And so what we have done with this Chang-2 system is designed it so that the analysis layer, so like the computational analyses that are being run, are independent of the interface that the user interacts with when they're trying to work with that software. And so what that means is that we can tailor different interfaces for different users with different levels of computational sophistication. And so, for example, if you have, say, like a domain scientist, like a cancer researcher, we have graphical interfaces that they can work with. And so they don't have to worry about things like working with command line software or logging into the cluster directly to run an analysis. But we also are trying to support data scientists, for example, like who may be building complex pipelines to automate data analysis, say at a DNA sequencing center. And so those users have other options that are going to be more convenient for them to work with and easier to automate. And so things like you can access this from the Python programming language or from a command line interface. And we are also building some functionality that helps keep that lab notebook that I was describing earlier, helps keep that for the researcher. And so like as you're working through an analysis with Chang-2, behind the scenes, it is tracking every single step that you ran for you, and it's keeping very detailed logs of that information. And so I mention this here because I think that as this field continues to progress and develop, and as we get more folks who are sort of trained as software engineers, but with a background in biology as well, working in this field, these kinds of things are going to become more and more standard. And so the software will continue to get easier to use, the interfaces will continue to get more accessible, and the methods will continue to get more reproducible, ideally by doing things like this, where the software itself is tracking what it's doing for you. Now, I mentioned that a lot goes into like building and distributing a software project, so that you may not necessarily want to jump into that if you don't have to. A really good example of that is the user support forum that my team runs. And so we have a site that we use to provide technical support to users of our system. We call this the Chang-2 forum. We have hundreds of people visiting this a day. We have tens of new questions being posted every day. And it is a community where users are helping users, but we have invested a lot of time in moderating that. We spend a lot of time answering questions on there. And that has really, I think, more than anything else, the Chime 2 forum has like enabled me to get funding for this project. So not that we've built the software, but that we are very actively engaged with the community in helping people use that software. Similarly, I mentioned workshops and getting involved in a workshop if there's some software that you're interested in, or just if you want to start developing more sophisticated computational skills. My team runs a lot of workshops. These are another thing that I think has really like enabled us to be able to bring in good funding for the project through our engagement with the community. But what we do in these, and you'll find this at to varying degrees at different workshops that you go to, but like these are interactive workshops. And so you can see this is one of my PhD students up here teaching. He's doing something on his screen, and all of these folks out here are following along. And this is a very hands-on event. And I mentioned like networking is common. So like we have poster sessions for the attendees to present at. We do small group sessions so that we can help people with more specific questions. And so if you've never been to this type of a workshop before, I highly recommend it. I mentioned we move these online. This has been a very interesting learning experience. We did a workshop back in October that was funded by the Chan Zuckerberg Initiative. And we were able to offer this workshop at a very low cost thanks to the grant funding for it. But we ended up having about 75 participants in this workshop who joined us from all over the world. So I think we had something like 35 different countries represented. And so this was really a super exciting thing for us because it was really an opportunity that was created by everybody having restricted travel due to the pandemic, but we were able to reach a lot of people who we typically would not have been able to reach because we did this online. And so we had a lot of folks joining who typically would not be able to say like travel to the US or travel to Europe or wherever we're doing one of our workshops. But because we were doing this online, they were able to attend from their home. And so it was a really cool experience. And we are definitely going to be continuing to do these online workshops after the pandemic. We will be doing some in-person ones, I imagine, but I think online offerings is going to be something that we carry forward. And I suspect I've spoken with other folks who have run these types of workshops since the pandemic. And I think this is something that we are just going to see more of afterwards because everybody has realized that there are a lot of benefits to doing these online in terms of reaching a very diverse community, lowering the carbon footprint of the event because, we're not all flying all over the place to go do this, and just making it accessible to people who otherwise would have a hard time attending. Okay, so I want to wrap up here just in the next few minutes. Actually, before I do that, do we have any questions at this point?
I don't see any in the chat. Anyone? Just holler out. Okay. Okay,
I'll just keep going then. So I'll probably just go up until, I think we have till 3:30, right, Jani?
Yes. Yeah.
Okay, so I'll probably just go, for another 7 minutes or so here and leave another couple minutes at the end for questions in case anybody's saving them up for
throw something in the chat if you think of something. So.
Okay, so In my workshops, I often start with talking about the beginning of microbial research, which I usually think of as starting with Anton van Leeuwenhoek, who is credited with essentially with the discovery of microbes, because he was able to create some very powerful microscopes in the late 1600s, and was basically pointed these microscopes at everything that he could imagine, from dental plaque to seawater to soil, and described and documented all the little critters that he was seeing in these environments that he was looking at. And he sort of famously referred to these as animalcules. And so this is really where my field of microbial research got started. The reason I bring this up now is I mentioned that I wanted to talk about open science a little bit, and something that is a little bit less known about Leeuwenhoek is that throughout his life, he did not give any descriptions of how he created the microscopes. He wouldn't tell anybody how he did these, how he built these. And so he would show people, so he would invite people to look through his microscopes and observe these organisms that he was seeing. And he would share his findings, but he didn't tell anybody how he created those microscopes. And so when he died in 1723, the microbial world basically faded from view for about 200 years. And so it wasn't until compound microscopes were invented a couple hundred years later that people were able to start seeing these organisms again. And in fact, in that intervening 200 years, some people even called into question whether these things really existed that he had described. And so, that is sort of, I'm not going to criticize what he was doing. It was a very different time, obviously. And as, the author of this article notes here, we have hardly resolved the issue of intellectual property since then. So I'm not trying to criticize, but it makes me wonder where would we be in this field right now? How much better might we be at controlling pandemics, for example, if we had an additional 200 years of microbial research behind us? And so, my opinion is that we face a lot of global problems right now. And by being as open as possible with our science and with our data and with our methods, that enables us to work together to solve those global problems. And so, as anybody who knows me or has worked with me knows, I'm a big advocate of trying to get data out there, trying to get software out there, and make it open and accessible. And with my software projects, for example, that Chime system that I've been telling you about, that is completely open source and free. So anybody can download that, including commercial organizations. They can actually adapt it if they want to. And make their own versions of it. They can audit every single line of code. And so you could go on the internet right now and you could find exactly what's happening inside that software. Similar with data, and there's definitely times when this is not possible for privacy reasons. But with data, same idea. If we can be as open as possible, that creates the possibility for future researchers to integrate, say, our work in a meta-analysis that might solve some problem or might shed some light on some new area of science. And it enables us to have an even bigger impact with the research that we're doing. Oops. And so I'm going to wrap up with another few tips here on open science and just how you can share what you're doing with the research community in the most open way possible. First thing that I like to mention here is publish in open access journals. Sometimes that's not possible. We all know that the costs can be very high, publication costs. So, I mean, sometimes thousands of dollars. So if it's not feasible, a workaround for that is to post your work on preprint servers. So something like bioRxiv or medRxiv. That is typically, for most journals, that is something that you're allowed to do, and they don't consider that prior publication of the work. And if you make modifications to that work, say like through the peer review process, you're allowed to update that preprint on the server. And so if your paper goes into, say, a closed access journal and it's behind a paywall, people who can't afford to or don't have the journal subscriptions to access that work could still access the preprint. And maybe it's not the final version of it, but it's probably pretty close. And so it's kind of a hack to get your papers out there for free, to be read for free, even if you can't afford the open access fees. I mentioned like bioinformatics methods earlier. So like keeping track of all the successful commands that you ran. I like to think of that as showing your work. And that is definitely part of doing open science, is presenting very detailed records. I also talked about data deposition. And so by depositing your data and your standards compliant metadata in public archives, that is a great way to make your work have a bigger reach, have a bigger impact, because others will get that, and they will, in many cases, often end up using that in meta-analyses down the road. If your data is in one of these resources, another benefit that you get is that when somebody comes to you a few years down the road and says, hey, can I get the data that you used in that study? You don't have to go dig through backup hard drives or email the postdoc who left your lab 4 years ago to figure out where that data is. You just say, oh, it's in the NCBI sequence read archive under this accession number. And that's kind of it. You can then just point them toward that resource. If they have any technical challenges accessing the data, it's not your problem anymore. It's the database's problem. Pre-registering your study is an interesting idea that's becoming more common now. I've got a link in here, and I'll share the slides with Jani afterwards so she can pass them around to everybody so you can access these links. But this is basically a way to describe what your hypotheses are before you actually carry out your study. And in my opinion, that's really good practice. It's an extra step for sure, but it does, support you when, you find something from your data. It helps to support your finding if you can say that this was a hypothesis that I had registered before I started analyzing these data. As our data sets get more and more complex, it is tempting to either purposefully or inadvertently mine that data, and essentially what you end up doing is developing and testing hypotheses at the same time. Other things, teach, like those workshops. If you have some skills that other people can learn, it's getting really easy to do this now with all the remote workshops that everybody is doing. And if you're developing code, definitely try and release it under open source licenses. Like I mentioned, that's the situation with Chime. It's totally free. Anybody can use it or read it. And so I will wrap up there. I just want to put my acknowledgement slide up here. And in particular, I want to thank the Partnership for Native American Cancer Prevention. I have been involved with NACP in one form or another, probably for 6 or 7, 8 years now, maybe. And they have been extremely supportive of me as I've been developing my career at NAU. And so I just want to thank Janny and the whole NACP team for their support of my work. And thank you so much for your time. I realize we're right at the end now, so I'll take a question if we have them, but you can also find me by email pretty easily.
Yeah, so is there any questions? So please don't leave us if you were participating, to learn more about bioinformatics. Kelly Lorela, our evaluator, has put a link for some feedback so we can, continue to make these workshops as good as we possibly can. The other thing is that we are working on, Ron and I and Sheena and Rhea Pravat, who are the GUIDES team, a website really for more resources for everyone associated with GUIDES. And so, Greg, I was thinking it would be great if we probably will, well, we'll definitely put you as a resource, but also perhaps a link to the paper that you talked about, and then maybe to your website, so that, if someone was interested in actually getting the Chime software, that type of thing. So we could talk a little bit more about that, but we will definitely make that more available or easier to access. And then, like he said, he'll send me the PowerPoint, and we'll, send it out to everyone who is here, and perhaps even make that available on the website. So, but yeah, we sure appreciate your time. Very interesting. I saw so many parallels to just analytical chemistry, which is my background, and particularly this, let's write everything we have so that people can, learn from it, that as opposed to skipping a few steps and then you can't reproduce data, that type of thing. So
yeah,
really interesting. I guess the last thing I would say is that if you are, as long as it's connected to an NACP project or something that you've done, maybe a student who worked on it, and you're having a little trouble making that open access cost, let us know as leadership of NACP, and we can see if there's a way that we can help pay for that. Because I know I agree with Greg. It's kind of expensive, but when you go to look for papers, it's so much nicer just to be able to get it easily, as opposed to trying to figure out, how can I, ask someone to send me a PDF of this. So. Any questions? Anything else for Greg?
Sure, Ron has a question.
Yeah.
I just have to ask something. So I'm curious about how you think about the projects that you're doing. And conceptually, I'm thinking about a project and try to determine how a microbiome affects or interacts with the epithelium that's surrounding it. And in the epithelium, I'd like to know transcripts and then metabolome. So my question is, how do you conceptually formulate that question and then begin to lay out the pieces that you would carry out?
Right. Well, I guess, I think about it in more or less like the same way I would think about, just a typical experimental design. I would want to think, very specifically about what type of information I'm interested in. And what you're describing, it sounds like it's probably like a multi-omics kind of survey.
Yes.
I think you had mentioned RNA-seq and metabolome
Yes.
data. And so it sounds like you're clearly on the right track there and thinking about like, okay, what are the data types that I'm going to need here to answer the specific questions that I'm interested in. And so like, I'll usually think about, like when I'm either designing a new project myself or with a collaborator, I try and think about it in terms of like that layers idea, like which of the layers are going to be important for this particular project. Like, do I need to know what bacteria and what fungi are there, or do I just need the bacteria in this study, but I want to know, what metabolites are in that environment? And so I just start thinking about like piecing those together. And then of course, you've got to balance that with the budget, because like, sure, it'd probably always be nice to know what fungi are there too, but maybe that's, beyond what you've got for this project. And then, the thing that is often gets very complicated, especially in like the human studies with these very high dimensional data types like microbiomes or proteomes, is that there's so much variation, like in the human gut microbiome across individuals. I mean, there's a lot that's consistent, but there's a lot that differs as well. And so you've got to be thinking about, is it possible to do a power analysis for this so I can figure out if I am going to have a big enough sample size? And like power analysis is really challenging in this area. And so like I'm often going and trying to figure out like, if I'm trying to figure out my sample size for a study, it's really common that I can't do like a formal power analysis the way, a statistician would have me do it, because I don't know what the effect size is of, say, this fiber prebiotic treatment on the gut microbiome. I have no idea what effect size I'm looking for. And so I'll start by looking for similar work, like what prebiotic surveys have been done in human subjects and what did their sample size look like. And in some cases, I've even done things like obtained public data sets and say like done subsampling from that to try and figure out, okay, like at what point does that effect disappear? And that can be sort of a more like experimental way to try and figure out like how many subjects do I actually need to carry out this kind of analysis. So, it's thinking about what are the data types that I most need and how does that intersect with what I can afford? And then it's moving on to like, how many subjects am I going to need to be able to carry this study out? Or, if we're working in mice or other lab animals, that tends to be a little bit easier because you factor out some of that variation that you see in the human population. So if these are all on the same diet and they're the same genetic background, then the variations in the microbiome tend to start decreasing quite a bit. So I hope that
really
answers.
Yeah. We just struggled through a grant and we went through all those different issues that you talked about, and sample size and power analysis just give me a total headache.
Mm-hmm. Yeah. Yep. And it's hard to do. I mean, power analyses are always hard because, you often don't know the effect size ahead of time. So,
yeah.
And,
I appreciate your answer. I mean, that really cements what we were thinking about.
Cool. I'm glad I was able to help a little bit. And, if folks are thinking about those kind of questions for their own projects, you know, I'm around. You can always reach me by email. Hopefully before too long, we may be able to get a cup of coffee and chat about it or whatever.
Right. Yeah, great. Great. And then I think it was Maria Pravat reminded me that we do have actually within the GUIDES resource core, we have some funds set aside for publications for our early stage investigators. So yeah, don't let that be a deterrent in terms of the journal you're choosing. So anyway, well, wonderful. Thank you so much, Greg. So it was great.
Yeah, my pleasure. Thanks for having me.
Yeah, and we will definitely provide you know, information on the website, which is sort of in the works right now, but hopefully we'll get at least a first draft up soon. So thank you.
Sounds great.
Right.
Bye, everybody.
Bye.
Yeah, thanks, Greg. That was great.
Yeah, my pleasure.